In this overview of meiosis, I want to describe the key features of this type of cell division that would eventually contribute not only to the making of the sex cells, organisms with a sexual life cycle and reproduce sexually rely on for the process of fertilization, but also how meiosis contributes to increasing the genetic diversity of organisms that reproduce sexually. Meiosis is going to reduce the number of chromosomes in the daughter cells produced in half. And for this reason, meiosis is often described as a type of reductional division. Meiosis begins with a cell that has the complete two sets of chromosomes you typically find in any body cell, but the daughter cells that are produced in the end are going to be haploid, meaning they only have half the number of chromosomes in the original cell that started the division process in meiosis. And that is going to be only one set of chromosomes, or haploid. In this illustration, you can see that some of the terminology we've learned previously about mitosis is going to repeat. There's a prophase, a metaphase, an anaphase, and telophase. But notice how these are going to be repeating on a second round of division. To be more specific about when something is happening, scientists have named the two types of division. We have meiosis I, where the main purpose is to separate the two homologous chromosomes of a pair, and meiosis II is going to be for the purpose of separating the two sister chromatids of a duplicated chromosome. Depending on which one of the two rounds of division you're looking at, a prophase would be called a prophase I if it's part of meiosis I, or a prophase II if it's part of meiosis II. And so the cycle continues similarly to how we saw the progression of these events in mitosis. Prophase I is followed by metaphase I, then we have anaphase I, then telophase I and cytokinesis. There is no interface between meiosis I and meiosis II. As soon as the two cells are finished being produced in meiosis I, they will immediately go into the next round of division, meiosis II, and continue with prophase II, metaphase II, anaphase II, and telophase II. Although there are a lot of similarities between mitosis and meiosis, there are also going to be key differences, and I want to talk about those differences so that you don't have any confusion as to which division does what. In this next slide, we are going to focus on the main events of meiosis I. And one of the things you're going to notice that doesn't happen in mitosis is that in meiosis, there's going to be pairing up of homologous chromosomes. If you can see my laser pointer over here, notice how there's like a long blue chromosome and a long red chromosome, because these two chromosomes resemble each other in size and shape, and also let's presume that they have the same type of genetic information. These two duplicated chromosomes are going to what we call homologous chromosomes. Notice also that we're specifying that one of these two duplicated chromosomes came from the father, and the other one in red came from the mother. So what happens in meiosis I, during prophase I, is that through mechanisms that are still the subject of much investigation, these chromosomes find their partner. The two homologous chromosomes pair up, forming a structure called a tetrad. You may have learned in mathematics that the prefix tetra or the word tetra means 4. And so there's something here that adds up to 4. If you look at the blue chromosome, it's made up of 2 sister chromatids. Plus, look at the red chromosome, and it's made up of 2 long red chromatids. 2 plus 2 equals 4. In this tetrad, in this combination of the 2 homologous chromosomes, there's going to be a total of 4 chromatids. Next to them, you're going to notice that there is a pair of smaller chromosomes. So there's a short red and a short blue chromosome, And these also consist of each two sister chromatids. So we have like a tetrad with the longer chromosomes, and we have another tetrad with the shorter chromosomes. And here in this cell, we're only showing 4 chromosomes for the sake of simplicity. When these homologous chromosomes pair up, they also engage in an exchange of genetic information, which I will be describing in an upcoming slide. What follows after prophase one is going to be metaphase one. Notice that in metaphase one, the pairing of the homologous chromosomes continues, and that is exactly how they line up along the metaphase plate. Here's our imaginary metaphase plate. It's usually right around the middle of the cell, and here we can see that the long blue and the long red chromosome are still next to each other, and that the short red and short blue chromosome are also still together. As you can expect, when anaphase 1 comes, there's going to be a separation of genetic information. But look at what is being separated. The two chromatids of the blue chromosome are still together. What is separating here is going to be the two homologous chromosomes. One of each type will go into an opposite side of the cell. So the long blue chromosome goes to the left, the long red chromosome goes to the right. Keep in mind that this was a chromosome inherited from the father, and this was a chromosome inherited from the mother. The same thing happens for all of the other pairs of homologous chromosomes in the cell. The two homologous chromosomes that were smaller in size are also being separated. One is being pulled to the left, and another one's being pulled to the right. After anaphase finishes the process of separating the two homologous chromosomes, there will be a telophase. And notice what is happening now. You can see here that in this cell, there's going to be only 2 chromosomes. However, let's go back to the original cell we began with. This cell has 4 chromosomes to begin with. So this reduction from 4 chromosomes to 2 chromosomes is exactly what meiosis is going to do. The products of meiosis, the daughter cells made by meiosis, are going to always contain only half of the number of chromosomes found in the original cell that began the process of meiosis. Let's focus for a moment on the exchange of genetic information that I said takes place in the prophase 1. Here you see once again a tetrad. So we have a red chromosome that was inherited from one of the parents, the blue one inherited from the other parent. You can also see in these chromosomes, we have marked the location of specific genes that are carried within the chromosomes. So this capital C, if you look at the lower left corner of the screen, my illustration, the capital C will be a section of DNA, a gene responsible for producing brown coats in mice. If you look at the capital E, I'm moving back to the upper right corner, this capital E location on this red chromosome is going to be for the section of DNA that is going to be coding for the production of black eyes in mice. Then, if we look at the blue chromosome, there's also a little lowercase c and a lowercase e. Those are the alleles or genes that are going to be responsible for the making of white coat and pink eyes in mice. So this would be the chromosomes that are expected to be present in a mouse that was the result of the sexual reproduction between a brown coat, black eye mouse, and a white coat, pink eye mouse. What happens when this mouse is going through the offspring of the 2 original parents, when they go through the process of meiosis, is that these 2 chromosomes, the homologous chromosomes, are going to exchange portions of their DNA. So you can see here that there's a small portion of the red chromatid, one of the 2 chromatids in the red chromosome, and a corresponding piece in size and the type of genetic information from this blue chromatid. These 2 are going to be detaching at a specific point, and they are going to be trading places. When all of this is happening, the 2 chromosomes are being secured, held together by special proteins called bindings. Then, when these pieces become clipped, instead of rejoining back the original chromatid they were once part of, they are going to be attaching to the corresponding or the adjacent chromatid that is next to them. This exchange, this crossing of the two chromatids is what we call the chiasma that leads eventually to the crossing over of information of chromosomes. Then the outcome is going to be this. Look at this chromatid that I'm showing here with my laser pointer. This chromatid used to be all red and had brown coat and used to be next to an allele for black eyes, but not anymore. Because of crossing over, this chromatid now has genetic information for producing a mouse with brown coat and pink eyes. Likewise, take a look at the chromatid just next to it, the one with which the exchange of crossing over took place. There is a chromatid over here that has the gene for white coat. Next to it, there used to be a gene for pink eyes, but not anymore. The gene that is going to be next to it is going to be a gene for black eyes. And so now it is possible to produce offspring with different characteristics from what we saw before in the parents. Offspring inheriting this chromatid will now have brown coats and pink eyes, something that neither of the original two parents displayed. Offspring receiving this chromatid are going to have white coat and black eyes. And that is, again, something we didn't see among the original parents that we saw that produced the offspring we are now describing. So when you see these chromatids that have exchanged genetic information, we call these recombinant chromosomes because there's a new combination of alleles within the chromosome. One important detail to remember, and you can expect that I'll be asking questions about this in the exam, is that when these events are taking place, so crossing over, something that increases genetic diversity, takes place in the prophase I of meiosis I, of course. In metaphase I, we're still in meiosis I, there is another interesting event that takes place. When homologous chromosomes are paired up, and you can see here we have a tetrad, the one consisting of the longer chromosomes. Here we have another tetrad, the one that is consisting of the shorter chromosomes. When they line up at the metaphase plate, when these tetrads line up at the metaphase plate, they are going to line up in any way, in any order they wish to do. So, for example, we can have all of the blue chromosomes facing to the left and all of the red chromosomes facing to the right. A question somebody may ask in class is this the way this is always going to happen? And the answer is absolutely not. There are any number of possibilities that can take place. Instead of all blue chromosomes facing to the left and all red facing to the right, we can have the long blue and the short red facing to the left, and we can have the long red and the short blue facing to the right. The outcome is going to be that many combinations of paternal and maternal chromosomes can exist in the cells that are meeting meiosis. With this simple example, we're beginning with only 4 chromosomes, 2 blue and 2 red. There's a possibility of up to 4 different combinations of chromosomes in the daughter cells made by meiosis. If you were to think about human cells, which instead of having only 2 pairs of chromosomes, they have 23 pairs of chromosomes, the possibilities are going to be much greater. Just because of independent assortment alone, the number of combinations can be up to 8 million within each of the within each of the organisms, each one of the humans that is responsible for the making of the sex cells. So these are the things that are happening in meiosis I. Remember, here in the prophase I, when the tetrads are forming, when homologous chromosomes are finding their partners, there is going to be crossing over. And in metaphase I, when tetrads line up along the metaphase plate, they do so in a fashion that is completely independent of other tetrads and what they have done with regards to their orientation. Meiosis I then produces 2 cells that are going to contain only half the number of chromosomes, and they will be completely genetically different from the parent cell. To finish the process of meiosis, we have to go through meiosis II. And meiosis II is going to be a lot more similar to mitosis, except that we're only beginning with half the number of chromosomes in each of the cells that are about to undergo meiosis II. Remember, we began with a cell that had 4 chromosomes in the nucleus, but now we're beginning with cells that only have half the number of chromosomes, and that is going to be 2 chromosomes in their nuclei. The reason meiosis II is important is because no cell division, listen carefully, no cell division can be finished and still have chromosomes consisting of 2 sister chromatids. So if you look at the chromosomes in the cells at the beginning of meiosis II, each still has the 2 sister chromatids that were made during the interphase stage and the duplication of the chromosomes. Meiosis II is needed to separate these sister chromatids. So from the prophase, we go to a metaphase where chromosomes line up independently. We have no homologous chromosomes here anymore because the homologous chromosomes have been already separated during meiosis I. But now anaphase II is going to be responsible for separating the 2 sister chromatids of each chromosome. The result is going to be cells that each will contain 2 chromatids, and those chromatids now are chromosomes in their own right. And so these cells that you see here at the end of telophase II are each going to be containing 2 chromosomes each. Notice also that because of meiosis II, dividing 2 cells in half, now we're going to end up with a total number of 4 daughter cells that are being produced. And what are these cells going to be used for? They're going to be used for the purpose of sexual reproduction. These are going to be eventually maturing to the gametes, the sex cells, eggs and sperm, that are going to be used during fertilization. And because of the events of crossing over and independent assortment, these cells are going to be completely genetically different from the original cell that began the process of meiosis, and that in turn increases genetic diversity in any species that uses a sexual reproductive life cycle to produce the next generation.